With that being said, we'll go ahead and turn it over to Dr. Lauren Dypenbrough with the University of Florida. She'll be talking to us about the different strategies and management of arthropods with during this time of HLB. I know that's a concern and thought on everybody's mind, especially with it being found here in Georgia on the coast. Without further ado, you are.
Good morning, everyone. So today I'm going to give you some updates on some research that we're doing in Florida that I think is going to be pertinent up here as you're now experiencing everybody's favorite pest, Asian citrus psyllid, and the unfortunate greening that can come with it. I'm also going to talk about some of the other pests that are starting to emerge. And the speaker right after me is sick, so we can take more time. If you have questions about anything, you can feel free to interrupt. It doesn't bother me if you have a burning question that pops up. There's a lot of bugs, and I'm only going to hit a couple of them. So if I didn't get your favorite, I'm here all day. Just 3 o'clock, I have to give another talk, so that won't work. All right, so the majority of my focus today is going to be on Asian citrus psyllid management research, a new study that we've been working on for a couple of years now. We actually are at the point where we have data, we can say some things. So some of the data is mine, some of the data are coming from a pathology lab, because I figured folks would want to know what the impact of some of the stuff is on Huanglong Bing. Some information on mite management. Mites have become a bigger challenge lately in Florida, and Josh and I were just talking about some mite issues up here. We're doing some more research. Never in my life thought I'd work on mites, but here we are. And I have a mite specialist in my lab, so I'm taking full advantage of it. And then some new and resurging pest challenges. Citrus is still fairly young in Georgia, so a lot of these pests are new no matter what for y'all, but we do have some new stuff in Florida to keep an eye out for. All right, so Asian citrus psyllid. I'm just gonna go through a little bit of information about it and HLB for those of you who have not yet had the extreme education experience that we've had in Florida. So it's been in Florida since 1998. It's now endemic through the majority of our growing regions. It's not heavily abundant in North Florida. It is there, but not super abundant. And now it's found in Georgia. Sorry, we didn't really want to share with you guys, but it happens. It is the vector for Candidatus Liberibacter asiaticus, which is CLAS, and which is a phloem-limited bacteria, so it stays entirely in the phloem of the plant. It's also called Huanglong Bing, HLB, citrus greening. We have lots of names. So most likely I'll flip between HLB and citrus greening. They're the same thing. So the bug itself causes some damage from its feeding, which includes dieback at new growth. So you'll get heavy infestation at growth terminals, new flush. They'll actually die back and look kind of black and withered. You can get witch's broom in the canopy from heavy feeding, which is the result of that initial dieback. In Florida, they're present pretty much year-round because we have hosts available year-round. With citrus greening, you get off-season flush. So instead of the main 2 flush periods and sometimes a 3rd extra flush period we used to have, we now have flush throughout the year. And so they can stick around year-long. This is one of the problems with HLB is that causing that off-season flush maintains those populations. So if you guys are still young in this cycle, you're not going to have that crazy off-season flush like we have. So if you can focus on managing in your flush periods, which I'll talk about a little bit later, you should be able to hopefully keep this at bay a little bit. Citrus greening. Again, I know this is just a new find up here. It's probably just literally somebody has a titer, has a little bit of damage. So this is a bacterial disease of the vascular system, and these are some of the symptoms. This is what we call blotchy mottle. If you look across this, you can see how the yellowing is kind of not matched up on either side of the midvein, and so you have a little bit of yellow and weird spots here and there. So it's not evenly dispersed like you would see if it was like a nutrient deficiency, it's just random. We also get these corky veins, which are these thick veins that just stick out. The first time I saw that in the field, it was pretty shocking to me because I'd never seen it before 2018. And then the infected trees have really gross looking fruit. They don't green up right. They look kind of yellow, and they don't taste right. They're very bitter. If you see, they're lopsided. If you were to cut through the middle, you'd have this weird little lopsided thing. The seeds get aborted. The central core is kind of curved, and you get this weird staining pattern. But bigger issue, you can't sell this fruit. Nobody wants to buy a green fruit at the store. Nobody wants to eat a bitter citrus fruit. It's not what we want. There's other symptoms listed up here as well. There is some really nice research that's happening in Florida looking at flavor additives for juice fruit. and nutritional additives just to keep your trees healthy enough to produce some decent fruit. We are actually still producing some nice fruit. That is not my area of expertise, but if you're interested in that stuff, I could put you in contact with the folks that do that work. So the good news is there is a natural enemy for Asian citrus psyllid. There's lots of things that will eat them. Lady beetles will eat them, lacewing larvae will eat them, syrphids, which are flies, their juveniles are predatory, spiders, minute pirate bugs, ants, and parasitoids. Ants also have a weird relationship with them where they'll protect them from predators. So if you have a lot of ants and you want to support predators in your system, ant management should be part of your management program. This is Tamarixia radiata. This was an imported parasitoid that's quite effective. So here's your adult, and she finds a host. She lays the egg. It's kind of on the side of the host. The egg is right here. I'm sorry, these pictures are really, really old, so they're a little grainy. There's the egg there, and then the larva develops on the underside and basically makes a mummy and pops out. You'll see like a hole out of the top of the pupa that's dried out. Unfortunately, even though this is present and it's very effective where we can establish it, we spray a lot of insecticides, and so they really can't take hold and do their job because this is a pathogen vector. So we really have to worry about management of it. You could possibly get this incorporated while they're still at a low density, but you're just gonna have to inundate your fields. And the Florida Department of Agriculture does do some rearing of this parasite, so if folks are interested in that, the laboratory in Dundee, Florida is the place to get those parasites. I think they might ship up here, I don't know. So a little bit of our history with HLB and management. From 1998 to 2018, we did intense Asian citrus psyllid chemical management. We're talking like every month minimum, 12 to 14 sprays a year to try to keep this pest under control. And that was because they knew what the pest could do and what it did as a vector. And these included neonicotinoid drenches for young tree protection, calendar sprays for psyllids. They sprayed the heck out of the system, but it didn't work in the long term. It was a crutch. It gave us some time, but it didn't really work out. These were part of what we called citrus health management areas, which is an area-wide management program. And one of our biggest challenges here was that it required growers to work together. And that didn't really work in Florida. So some of the bigger growers would spray everything, but then their neighbor next door may not be able to afford all the chemistries. And so you'd get these reservoirs of insects. Any type of area-wide management program requires entire buy-in from everybody. And it just didn't work out. So it's also very hard to do when you're on the mainland. Islands often are more successful at this. Hawaii has done some really nice area-wide management programs for fruit flies in particular. So the goal with this, though, is to reduce the population by minimizing movement between groves. These are a highly mobile adult, and slowing the development of pesticide resistance by making sure everybody was rotating those chemistries. So the CHIMAs would coordinate these. There was usually a large grower in the area that coordinated everything, or one of the bigger industries, and it was supported through the University of Florida. And this is all well before my time of getting there, but it worked for a while, and then it didn't work so well. And you needed to have that full buy-in to get that economic return. So where we are right now, most of our citrus management is going to be for Asian citrus psyllid. We have some other pests in the system that are pretty bad, and if you have them, you're definitely managing for them. But the majority of the chemistries we apply are focused on Asian citrus psyllid. Now, the number of sprays and what we spray is going to depend on the market and the individual grower choice. If you're a fresh market producer, you're definitely going to be spraying a lot more than a juice fruit producer because we can handle a little bit of damage. We know how to process the fruits. We know what additives to use and what mixtures, what blends of fruit actually tastes good when mixed together. Fresh market, as you all know, satsuma up here is a fresh market item, not tolerant of any imperfections, so you have to keep those trees really clean, which is a huge challenge. Most of my growers are actually juice market producers, and we know we have psyllids. We know we have them, so we try to keep them at bay as best we can, but they're gonna be there, and it does affect our juice quality, so it is something that they work on at a larger scale. For the HLB affected trees, which is most of the trees in Florida, we know that nutrition is really important in keeping these trees productive. Tripti Bashis and Jude Grosser have both done a bunch of work looking at nutrition. And we actually have a large nutrition program going in the state of Florida right now where growers are cooperating with the university, sending soil samples and leaf samples and getting a literally grove by grove nutrition management program to help them actually have decent trees. It's a lot of work, but it's not impossible. And then these trees will receive a combination of foliar feeding and soil applied fertilizers. And why we do that combination is that as greening courses through the tree, you actually lose roots. And so it kills off roots eventually, and then the roots cannot absorb all the nutrients. So you're gonna have to put some to the foliar, some to the ground, and between the two, the trees are actually getting enough nutrition to survive and produce some fruit. All right, so I'm gonna flip into some of our current research, especially one of the things that we are worried about in Florida is that we have all these old groves. We're pushing groves, we're burning them, and then we wanna reset, but can we reset, and is it economically viable? So we have a team of researchers who are looking at some of these options. And so that kind of falls in place here with these targeted management programs. So you need to think about when your populations are very vulnerable. So lucky Georgia, you have an entomologist now in citrus. And so Arthropod is going to be learning the cycles of, and if you haven't met him, he's right there, by the way. He'll be learning when your psyllids are abundant and active. And if you target your sprays towards when the juveniles are most abundant, you'll get the most bang for your buck with your sprays. Adults move, juveniles can't fly. Hit the juveniles, hit the eggs. Managing multiple pests at once is a big issue. Historically, we've always looked at it as like, oh, I'm just working on this bug, I'm just working on this bug as scientists, but no grower functions like that. And so there's been a big shift in thinking about how we can manage multiple pests at once. And when I say pests, I mean insects and pathogens. So are there things we can do combined that hit both? Incorporating some of our non-insecticidal management options, and that's a big thing for resets, and I'll show you that in a minute. And then looking at how we can reduce our insecticidal inputs. These are getting more and more expensive. As our growers are focusing on nutrition, they're using trunk injections and other tools. We need to think about the economics of this and thinking about what is feasible. So for your targeted management, we know that Asian citrus psyllid are attracted to fresh flesh, fresh foliage. The adults can feed actually on the hardened leaves and the soft leaves, but they only mate and reproduce on flesh. So you really want to target your flush periods. Like I said, the nymphs can't fly. They're stuck there. So if you spray something, they're getting a full dose of it. A lot of our management now is targeted towards the flush periods, and that should make your best impact as far as killing the vectors. And then, as I said when I started, some of our big challenges. HLB causes the random off-season flushing cycles. And so now what do? You have like 6 different periods of flush during the year. You got to move your equipment field to field to field because we work with really big farms. And so some of them kind of have a schedule of we know this time of year is about when we have flush, and so they get all their sprays scheduled really tightly around there. And it's always a juggling act. And then the other thing to consider is if you're really focusing on the nutrition for your trees, if you're going to be putting a lot of nitrogen into the soil or into your tree system, insects love nitrogen. If you want to flare an insect population, put out a ton of nitrogen. You need it for your trees, but if you go too high and it flushes too hard, you are going to be battling every piercing, sucking insect under the sun. And I'm talking raining honeydew from scales. And I've experienced that one from somebody else's trial. It was not pretty. And that includes psyllids. And then the targeted management, we think it can work. There's some research right now going on where they're looking at plant growth regulators to actually try to better time the flush and also better time your fruit development periods. And that's work that's happening in Tripti Veshi's lab. And so we're kind of coming in on top of that and looking at, hey, can we actually manage the flush periods once she has the PGRs all figured out? And her research is really nice. If you haven't had a chance to check it out yet, a lot of her talks are on the CREC website. All right, so some of the research I wanted to show you, I kind of jumped ahead earlier because I thought it was the next slide, off in my head a little. We're reading a little bit about what citrus insect and mite IPM is going to look like in this era of HLB, and in particular in our reset fields. We know that we can't spray this problem away. So insecticides are part of the equation, but they're not the entire equation. So we need to look at incorporating newer tools to develop better management programs, more robust management programs, and also a shift in the mindset for us as researchers that insect and mite management is part of a whole grove management program. You don't do this in isolation. You do it as part of everything else. And so we want to think about the impacts of these tools that we're testing on pathogen management, tree growth, irrigation, fertigation needs. And then as a side part of that, we actually tacked on a question of can we use similar tools in residential settings. In Florida for decades now, we've discouraged residents from having citrus trees, which in theory sounds good, but in practice that takes a lot of your voters away who actually care about agricultural stuff. Those of us who grew up in Florida had citrus trees in our yards, and we would love to have them again. I have them on my porch, but the psyllids can't get on my porch because it's screened. And so we really wanted to focus on not just the commercial side, but also making sure we're thinking about can we get stuff back into the residential side and support that side of Florida citrus. And I know that there are some people in Georgia that have their own satsuma at home, so this might be useful as well. So the tools that we're looking at are actually things that some of the growers started. And most of this research is actually driven by conversations with growers or some small pilot projects done in other laboratories. So reflective metallized mulch, that's the reflective stuff you see people putting down in vegetable fields. There was some work done in the I think it was like 2010, 2011, looking at the impacts of it on Asian citrus psyllid and HLB, and it was done at a really small scale, so you really couldn't extrapolate that up to a larger scale. Red dyed kaolin, previous research shows that it actually improves the health of mature trees. So what that is basically it's kaolin, and then we're adding that red dye that you use on mulch, and it makes it stick to the trees better. It also adds a little more shade. Most insects don't really see red, so we think it could be a little bit of an insect deterrent. Christopher Vincent is a tree physiologist, and he's had some really interesting research on the mature trees, so we wanted to see what they would do on the juvenile trees. Individual protective covers. If you haven't seen these, it's like a big mesh bag that goes over your tree. Lots of growers in Florida are using them, but there was very little research done on them. It's just like, hey, it's a big exclusion bag. It will keep your psyllids off. But there's other challenges there. Other stuff still gets in. So our goals also there were to look at that and see, what do we need to know about pest management with those? And then a comparison to a standard, which is a monthly insecticide application to protect the trees. But Cliff Notes version, that standard doesn't work. So here's what it looks like. That is the reflective mulch in a citrus grove. And it is not the funnest stuff to work with, and I hope I never have to work with it again because it was awful to lay and you get burnt from both sides when you're out there scouting. And then we also have this, what we call a reflective donut that we're looking at in residential settings to see if we could transfer that information into residential. And then in the commercial grove, we have our red dyed kaolin. As you can see, it's very dirty to apply, but it does stick to the trees quite well. One of the big challenges we have with it is that it doesn't stick to flush. So I'll come back to that when I talk about some of the data. And then we also are doing it in the residential setting. And you'll notice everybody's logo. This is from a series of videos that we did. These are the IPCs. These are actually very big IPCs. Most are individual protective covers. Most of the ones that you're going to see employed in the field are going to be 5 to 6 foot tall. And this was a project that we got an extension on our funding, so we wound up putting bigger bags on to see how long we could keep under the trees under the bags. I would never put an 8-foot bag on a tree if I didn't have to. After my experience so far, the 5 to 6 foot are ideal. The 8 foots are just a challenge as far as managing them. And then this is in the residential setting. And that's more the size of that one. So if I stand next to it, it's about my height. I can stand under the bag and it hits the ground. And then of course our standard. In the grove, we're just spraying our standard monthly insecticides, and then the residential side is way trickier because there's really not much labeled for use in residential settings outside of horticultural oils and soaps, and quite frankly, most of them don't work. All right, so there's a lot we don't know here. And this is why we did the study. We don't know the return on the investment, which stay tuned on that one. We're gonna have a full harvest in a couple weeks here, and then one more harvest next year. Arthropod and pathogen management. So how do we scout these things? Scouting in an open field is easy and, well, challenging, but doable. How do you scout those bags? Do you open the bags? Do you walk next to them and try to peek in? How do we do these things? What arthropods and pathogens will become problematic? You're changing the environment around a tree, you're changing what's on the tree, or are we gonna have other issues? And how do we prevent or treat these pathogen and arthropod outbreaks? And then what happens after the trees are too big for protection by these tools? So for example, the reflective mulch, it is an 8-foot wide piece of reflective mulch, and about a foot on each side is sunk into the ground. So now you have 6 feet. So in theory, the way it's supposed to work is that the light reflects and it confuses the insect and they can't find the host. All right, well, what happens when your canopy is too big for that? We don't know. We haven't gotten there yet, and I don't think those trees are going to get there. Each tool, then, like I said, they all through the growing environment, so you're going to have some differences in what's happening with each of these plants. And this is what our field looked like. This is actually one of our research centers. This was the day before we went on our COVID pause. We were actually planting everything. And the IPCs are out here, little baby trees. They look much bigger now. And so for our insect and mite scouting, and this is where I'll focus most of my stuff because my group does the insect and mite scouting, we're doing it every 2 weeks, aside from the winter period, at which point we do 1 time a month because there's just not much activity. We look at the number of flush per tree, because that number of flush is actually going to help explain some of the data, especially with your piercing sucking insects. Number of Asian citrus psyllid eggs and nymphs on the flush. Citrus leaf miner infestation on flush. Our mite populations, for us, we generally deal with rust and spider mites. I know you guys also have broad mites up here, but we really don't have much challenges with them, so they're not on our list. And I should note, for those of you who have heard me talk about Levick mealybug before, this project was designed before this thing showed up. And so it has been an incidental pest that we've learned about throughout it, and it has not been fun. And if you haven't heard this, I'm going to mention it later, and we could talk. And then we have a management plan, but sometimes we need to add extra management actions, and so we keep notes of what we're doing. All right, so flush counts. This is, again, one of those things, we think about this mostly for our Asian citrus psyllid and citrus leaf miner, because if your flush is going up, you're going to increase their populations. And so when you're looking across here, it's a little busy, but for the most part, your flush are still pretty well timed together. That mulch has a really high flush count. And actually, we had, I don't think I kept this slide in here, because we had so much flush and so much newly expanded leaves, we had citrus leaf miner like crazy, and then canker on those reflective mulch treatments. So for the leaf miner and canker challenge, we had to add some more treatments on those, and probably wasn't the best choice with those in the system. Your IPC doesn't flush quite as hard in the beginning, but it does flush quite a bit harder later. And then the kaolin's kind of in the middle with everybody going along. All right, so here's what we're actually interested in. What's the impact on the Asian citrus psyllid? Well, basically, this is your IPC. IPCs are the one, we had a hurricane that took off some of our bags, so we did get a little bit of psyllids here and there. But for the most part, they're really not in there. So if you're trying to keep psyllids off your trees, IPCs work really well. Just know that hurricanes sometimes blow them off. And then your reflective mulch and your kaolin, they all are gonna have obviously psyllids. They're open and available for colonization. And one of the big challenges that we had here that we didn't expect is that with the red-dyed kaolin, it sticks great to your mature leaves, but it does not stick to flush at all. So when we would go out there to scout, we would actually see bright green flush, like a little highlighter, little beacon to the psyllids. Come here. So not great in terms of that. It was very available to them. It was also very available to the leaf miners. So these are some data from Dr. Dudney's laboratory. She's the pathologist at my station. We've worked together quite a bit, and this is one of our shared projects. So there's 2 ways we're looking at HLB here. We're looking at the visual HLB symptoms, and then she goes back through and looks for the CLAS titers of the actual pathogen. And the reason we do this is that most people scout by visual symptoms, because you can't just go out and run a PCR every single day on your trees. That's just not reasonable. So in this trial, you'll notice there's actually an extra treatment on here. We wound up combining these after a while because reflective mulch without insecticide basically meant your trees are dead. They're gone. Every bug under the sun went to them. So you have your kaolin, or sorry, your control kaolin, IPC, reflective mulch, and reflective mulch plus insecticide, which are again combined in my slides because we had to for insect purposes. And what you see in here is that there's no symptoms for the first 15 months. Now, we'll say the first 3 months of that we weren't allowed to scout because we weren't allowed to be at the research station. So our trees were in the ground and we were just hoping they survived. And when we came back, we found very little blotchy mottle in those fields. And then they found less HLB symptoms in the reflective mulch for the first 2 years, but by December of 2022, it was nearly 100%. All the trees basically looked like they had HLB symptoms, mostly gonna be blotchy mottle. Some corking veins, and some of the trees get this weird offshoot of growth that's really hard to describe if you have not seen it in the field. In the IPCs, we start to see some symptoms in 2021 and 2023, and that's when they were still in the 5-foot bags. But as soon as we put them in the 8-foot bags, the symptoms disappeared, so we think those symptoms are actually distress symptoms. And we didn't know that the stress symptoms and HLB symptoms look so similar. And we also found, like I said, hurricanes blew our bags up a little bit. They put some holes in the mesh. You got these big thorns on your citrus trees, mesh going back and forth in a hurricane. You get holes, things get in. We do our best to check the bags at least once a month and glue them back together, staple them, replace them, do what we can. All right, so here's the pathogen. Trees that have the Liberibacter titers. So the first PCR detection was in December of 2020. And then there are fewer detections in the reflective mulch treatments initially, but they approach the same level as the control, which is nearly 100% in June 2022. And then the first PCR detection in the IPC from a low infestation level. And this compares to where your first visual symptoms are. So our visual symptoms appeared back here, and then the titers often take a little bit longer to show up. And again, these data are from Dr. Dewdney's laboratory. We're almost ready to publish this, but if anybody has questions about this later, I'll do my best to explain pathology, but I'm an entomologist, so bear with me. And then we want to know how much bacteria is actually in there. What do the titers look like? So the titers all start really low, but you start to see some separation of these treatments by mid-2021, where the control and kaolin trees have the greatest titers. So coming up here. And the reflective mulch actually had a reduced titer. And so there'd been some incidental data about this previously where they thought that maybe the reflective mulch would actually get so hot that it almost worked like the old thermotherapy units to actually kill some of the pathogen off. And then the IPCs delayed detection by 2.5 years, which the only reason that we had detection is that we had the bags blow up during some hurricanes or some holes occurring. And so, but even at that point, it still stayed really low. This is your IPC. So it really might be worth the investment if you want to keep them off your trees for at least establishment periods. All right, so some other questions. This is a little bit more visual data because the graphs just don't make sense. The visual is so much better. These are our treatments. So these are the 8-foot bags that the tree has basically filled into. We just took them off this fall because we didn't have much fruit set with these bigger bags. And so we want to give them a little bit of chill hours, which they're getting plenty of this week, in hopes that we'll have a nice flowering and fruit set for next season. And then these sad little things are the red-dyed kaolin. So you can see they are not doing so great. They have some issues. The titers are pretty high in them. We also saw a little bit of, I think it was iron toxicity in some of the leaves, and that was just from accumulation of the kaolin. This is your control. They don't look horrible, but I mean, these are 3 years in. These trees should be like this. They should be big. And this is your reflective mulch treatments. So they actually have a decent canopy. Not all of them look this good, but they actually have a decent canopy and they have a fruit set on them. Not perfect. It did take a little more input, some tweaking, but we have some potential here. So in terms of canopy development, IPCs have the most robust canopy, and this is from the physiology group, the equal canopy volume despite constrictions, the constriction of the bags. They still have a nice, thorough volume. They have a greater trunk girth, so they are putting on size in their trunk. And then followed in order as far as canopy and trunk girth is going to be your reflective mulch, which you can see from the picture, red-dyed kaolin, and the control. Yeah, that canopy just doesn't lie. The picture tells the story there. It's 2 times greater than even of the control. And so hopefully we'll have some nice harvest data where our first harvest should be, I think, next week, hopefully. So the general findings from this, the IPCs outperform the reflective mulch, the red-dyed kaolin, and the monthly insecticide applications for ACP and citrus leaf miner control. I didn't show you the leaf miner data, but basically the bags keep the leaf miner off. So if leaf miner is a challenge and you're putting in new trees, this might be a worthwhile investment for you for a couple years. Trees under IPCs did not flower as robustly as trees in the other treatments, and so that's why I said we removed the IPCs in November. And hopefully we'll have a nice flower and fruit set in 2024. And then the residential data, we're about 2 years behind on that one because that came from a different set of funding. And we didn't show the data, but they're very similar and on trend with what we've seen in the commercial planting. And so some of the future research areas that we're looking at is improved pest management under IPCs. One of the big challenges with IPCs is that other stuff gets in. So you get scales, you get every mealybug. You'd probably get cottony cushion scale. I would love it. Sorry. You get thrips occasionally. Chili thrips can pass through the mesh. Certain mites do very well in there, particularly citrus red mite. Greasy spot can build up in there. So there's some other challenges that have to be worked out to actually incorporating these into a program. But they are very promising, and most of our growers are investing in them if they're resetting their fields. One of the other big challenges is, how long do I keep a bag on? Is 6 months as good as 2 years or 1 year? We don't know. This is actually some stuff we've been trying to get funding to work on. And then management for susceptible trees once the IPCs come out. So we've taken those IPCs off those trees, and how long until they're so sick they don't produce? We don't know. So management for them going forward is going to be really important, at least until they tell me I can't afford my grove anymore, and then, well, then I'll have more data for you. Okay, so we're going to flip. I'm just going to give you a moment. Does anybody have any questions on what I just talked about before I move into the next topic?
So with the OTC treatments, assuming they do what people hope they do and you get more uniform flush, potentially, would that help break the cycle a little bit?
Yes. What we actually think might work, and this is one of the treatments that we put in the proposal, was when the IPCs come off, is to either be using the steroids or the OTC injections to try to synchronize flush cycle and also to try to protect the trees. There does seem to be a bit of a protectant built in with that OTC against the development of HLB. Yeah.
Once a tree's got the HLB, right?
Yes.
HLB is a bacteria, right? So it's throughout the tree.
Yes.
So now suppose like a leaf mite comes to the tree. Can he carry the HLB virus?
No. It has to be through the piercing sucking transmission, and Asian citrus psyllid in particular is very adept at acquiring it, and they have the right combination of things in their gut to actually proliferate the pathogen. It does quite well inside the insect, but no other insects in our system really that we know of can vector it. There's another psyllid out there, but we don't have it in the US, thank goodness.
So suppose like a stink bug.
Yeah, they can't vector it.
They roll up,
Nope. They'll cause some damage on your fruit, but they won't vector the pathogen.
they won't transfer the bacteria.
Nope. Only the Asian citrus psyllid in our system. So it is worth knowing what they look like. And now that you can actually have them here, I'm sure Perva can do a hands-on workshop and teach you guys how to scout for them. You can come visit me if you want to learn better. They're, yeah, and the pictures don't really do it justice because the adult looks so big in the pictures, but they're so incredibly tiny. And had I thought about it, I would have brought some in alcohol for you guys because now I can. Previously, I couldn't. And then the juveniles are very small. They're very confusing. People often think they're aphids, but they're definitely not aphids. But you wouldn't know that unless you look at aphids all the time. Yes?
Once you take your covers off the young trees, have you developed any approach to keeping those trees clean?
Well, I just took my covers off, and so most of the growers have switched into an insecticide regime as soon as they take the covers off. That's the next step, is we put in a proposal last year that unfortunately wasn't funded to look at, one, the timing of taking your bags off, and then two, keeping them clean once you take them off. Because you have this beautiful tree, and it's a great investment, and yeah, you're probably gonna get a nice crop the first year, but how many years are you gonna keep getting a nice crop? I grew up in Florida. I love citrus. It's delicious. And I would love to keep working in this crop for the rest of my career. So I'd really like to keep it around. So stay tuned. We are working to find money for that. And once we find the money, I will hopefully give you some answers to that question. Might be a few years. Okay, I'm going to switch over to some mite management because I know this is an ongoing challenge for all of us. So a lot of this is going to be identification, and there's some basic information on management. It needs to be updated. We are currently doing some research in my group right now, and there's much more that needs to be done, especially as far as resistance management goes. So know your mites. Rust mites are the first one we're going to talk about. So you have pink citrus rust mite and citrus rust mite. For all intents and purposes, they look the same. They're these little guys. This is way bigger than they actually are. If you scouted for them, you know they run too darn fast to really count. So if you haven't scouted for them, the way that I was taught to do it is take the shady area and turn it to the light, and they're going to start running. Count bodies that are running, because they're never going to stay still for you. And they're very fast. My friend told me they look like a bugle with legs. So if you haven't seen it, take bugle with legs, make it itty bitty. There's your rust mites. It's a great search. And so most of the time you're going to have citrus rust mite with a little bit of pink citrus rust mite mixed in. We manage them one and the same because same stuff works. Wedge-shaped, light yellow to pinkish color, 0.15 millimeters long. So they're super duper tiny. That's why I said if you're scouting, looking for running mites is easier than trying to find them being still. They feed on your fruit and leaves. And your severity is going to depend on population size and the time of the year in which your fruit are damaged. And so the population increases in dry weather, and like last year we didn't get much in terms of our summer rains. So our populations just went off the charts in Florida. I don't know what they looked like up here, but they were really bad in Florida. And then your likelihood of damage is going to increase. And so there's two types of damage that we see on our fruit. The fruit that are damaged earlier in their development, they get this what we call shark skin. The fruit's rough looking. It's this one right here. Sometimes a little more whiter than that, but it's rough. And the juice quality is not good. So fresh market, obviously you can't do anything with that, but if it's got that weird shark skinning, really rough, kind of, it turns a little whiter as the fruit matures, I can't even send that to a processor because it's not going to be good. And if it happens later in development, which we see a ton of, because a lot of early season management will have like Movento or abamectin in it that'll keep that down. Late season management, or late season, this is the damage that you're going to see. And we call this bronzing or russeting. And these fruits, they look bad, but they taste awesome, which sounds crazy, but actually that late damage actually increases the amount of sugars going to the fruit. So for our juice growers, a lot of them actually do want a little bit of damage later in the season, but for fresh, you just can't. So that is a challenge, but do know if you see these in your field and they're not going to fresh, you can still eat those. I would not eat that. It's not going to taste right. And rust mites are normally going to be in the outer canopy fruit, so they're a little easier to scout than some of your other organisms. Here's where some of your challenges are going to come in. They have a very short generation time, so egg to adult, 7 to 10 days. That's a week. So if you're spraying every week, that's a lot of sprays, and you're probably going to get some resistance happening. They have a high reproductive capacity. The adult females will do 2 eggs per day for about 15 days, and then that will keep going. And they have a pretty long life. They stick around for about 20 days or so if they're not managed. So you can get these massive population explosions without even knowing it. You just go out to the field and all of a sudden they are there and you're starting to see damage, and that's a problem. So some of the information, all the mite research here was done before I got here, and so we're realizing now there's actually, we think, a lot of resistance issues in our system that need to be addressed. We do know that they develop earlier on in the season with the new leaf growth. Usually this population is going to be dominated by pink citrus rust mite with a little bit of rust mite. Not that that matters for management, it's just kind of interesting. And then they'll move to the fruit when your fruit become available, and they'll start causing this damage. So again, old data, I inherited these. They need to be redone now that we've had some shifting in our temperature regime. But the pattern is going to be pretty similar. Where you get rust mite buildup, kind of late spring they start getting going, and I assume you guys will be on a similar pattern here, and you have that dry period, that really dry period, and they just go crazy. They go bonkers right in here. So if you're thinking about timing management, you probably need to get something in down in here, early May. And then they kind of go off, we get those seasonal rains around this period, July-ish, normally, not last summer, and they kind of drop off. And then they start coming back in the fall. And so if you've got fruit developing that you're going to pick for fresh market, you also need to think about managing in that October-ish period to try to knock them down so you don't have ugly fruit. You want to get as much money out of your fruit as you can, especially if you're growing Satsuma, because those are really good fruit. I hate to see them get tossed. So for rust mite sampling, and this information is all available with this page, in the Florida Citrus Production Guide, which is online. So I didn't bring a whole bunch of copies of them because they're heavy, but that's online. If you need a link to it, just let me know. You can download any of these PDFs with what we're doing. But for sampling, for processed fruit, we start in April. We look at the fruit and leaves, monitor every 2 to 3 weeks. In truth, most processors don't actually do that. They just time their sprays to when they know the populations normally are out. For fresh market, you do need to scout for these because you don't want to over-apply chemistries, getting resistance, and costing you a lot of money. We start in early April, look on your leaves and fruit, monitor every 10 to 14 days. When you start seeing that population build up, you need to start thinking about management. You're going to have random stops, and there's actually a chart in the last page of the mite management chapter that has a chart of how many stops for your acreage to help give you some guidance there. You're going to check at least 4 fruit per stop and look at about midway in the canopy. So don't go super high, don't go super low, just literally right in front of you. And like I said, I take the dark side of the fruit because that's where they're feeding, and I turn it and I watch them run away from the light because they hate light. And so that's the easiest way to find your rust mites and to get a good count of them. And they're going to move fast, so you got to be ready to count. And then we have action thresholds, and these are based on sweet oranges in Florida. Might be different in Satsuma, so just know that they might change as you get more and more research happening up here. And so for us, process 10 rust mites per lens field, so just your standard 2 centimeter squared lens field, and then fresh is 2 rust mites. It's because it's going to build up really fast, and you want to have nice looking fruit. And then there's some diagrams that are available. I think that one's not in the production guide, but it's just to give you an idea of your stops, your randomized stops. And you can make a fancy chart and do all the stuff where you can literally do random walks and have a certain amount of stops per acre, per 10 acres. All right, so then spider mites. I know y'all have some spider mites up here. We do too. Citrus red mite, Texas citrus mite, and 6-spotted spider mite are the 3 that we see the most in Florida. You might have more up here. There's a couple more species that we don't see very often. Citrus red mite in the past few years has really been problematic. They have gotten to some really alarming numbers. They're kind of cute though, you know, hairy little things. So your spider mites, again, increase during dry weather. This past summer with those very, very long periods of dry, I'm gonna go long, by the way. I figured you didn't care. Those periods of long, dry spouts, they really help their populations go. They feed on the upper surfaces of young hardened leaves, and they cause this damage that's kind of stippling. Their mouth parts are kind of like they poke and scrape a little bit. So you see a lot of little spots on your leaves. And then this is what the firing looks like. It kind of looks like the leaf has been burnt. So spider mite sampling, weekly or biweekly, just depending on your historical knowledge of your field. Sample unit's going to be mature leaf behind flush leaves. So find your flush leaves, go behind it, pick a mature leaf, and look there because they like the mature leaves. Sample 4 leaves per tree from 6 trees. And again, all of this stuff, the scouting information, is available in the citrus production guide in that mite chapter. And your threshold's going to vary based on your market, and there's a whole layout of the markets in there. The one thing to note when you're thinking about mites is that we have a lot of predatory mites, and they're really important. So we want to make sure that we're not overspraying for the pests, because we want to try to keep those predators in the system because they are going to keep them in check most of the year. So knowing who your mites are is really important, so you know when you're looking that you're actually seeing a spider mite and you're not seeing a predator, because they sometimes look pretty similar. And it took me years to start figuring out who was what, especially with certain species. So 5 to 10 spider mites per leaf. September to May is generally going to be your trigger to spray. And that's going to depend on your market. So the challenge with these guys is they will cause defoliation. So you don't want their populations to get too high, defoliate your tree, and then your tree can't photosynthesize, and it's just a sad, sad tree. So these, again, are from the Citrus Production Guide, and these are all recommendations for Florida. I don't think there was a mite chapter in the Georgia guide. I can't remember that. Do you remember? I don't remember seeing that. So these are just some recommendations based on previous research of when to apply and what to apply. And these miticides work on all your mites. So in the spring, thinking about Apta, Envitor, Movento, or Vendex. A lot of times in the spring, we put out Movento for other things like Lebec mealybugs are a real big problem in the spring when the fruit are setting. So we try to get a double duty there. Post bloom, you've got to set here. Summer, fall, and then if you need a supplement, and then always have an oil when you're applying. If you keep putting out I love 435 oil. It's cheap and does a pretty darn good job of smothering a lot of things, including many mites. So when I put out my applications, unless I specifically need a different adjuvant, I use 435 oil. Again, very cheap. I like it. I see somebody taking notes. I can send you these slides if you want, or links later. Okay, so there is one thing I want to note, because we have been getting a ton of calls about spider mites the last few months. Sulfur. Sulfur is used to kill rust mites. Does anybody know what it does for spider mites? It makes them go like crazy. It kills their predators. So watch your sulfur usage. Use it sparingly. Fall is a great time to put sulfur out, especially if you have rust mites. But do know that you're going to have to watch out for your spider mites after you put a sulfur application out. We have seen several growers in Central Florida who are very certain that they have resistance to certain chemistries, and having visited their fields, I would probably put money on them being right. And then they use sulfur to try to control the rust mites, and it works pretty darn good on rust mites, but it will flare your spider mites. So it is something to keep in mind. And that's actually a project that we just put a proposal in on, so we'd love to look at that a little more mechanistically to better understand it. Okay, so we got some few critters. So there's new/resurging pests. Do any of you guys recognize these pests? At least one of them you probably do. Diaprepes are jerks, and they're up here. All right, so Levick mealybug, which was that first one here, to the best of my knowledge, it is not in Georgia yet, but it will get here. So it is something to keep on your radar. This is Bulimulus bonariensis. This is a new snail. It is in Georgia. It is throughout most of the southeast, and I'm only going to briefly mention it because I have a whole talk on it this afternoon. So if you really want to learn about it, 3 o'clock in the veggie session, I'll tell you everything I know. And then Diaprepes are coming back. They are making a huge resurgence in Florida. All right, so Levick mealybug is, obviously it's a mealybug. It's invasive. It has been in Florida since, we found it in 2019. It causes quite a bit of damage, and it's spread throughout much of central Florida. We haven't seen as much migration into North Florida, but we're not looking that hard in North Florida right now because the damage is so bad in central Florida. Has a really wide host range, and I'm talking like, it loves citrus. Historically, that is one of its favorite crops throughout the world. Blueberries, ornamentals, you name it, papaya. Yeah, it really does a number on papaya. But it likes a lot of different fruit crops and a lot of different ornamental crops. And so we're seeing it expanded into some of the nurseries in Florida, and that is a challenge. We don't know what the long-term impacts are going to be, but we do know they can kill trees. We do know they can kill large portions of mature trees, too. The nymphs and adults excrete a lot of honeydew. When we first found this, the growers thought they had cottony cushion scale because we've had experience with cottony cushion scale in Florida. It is not cottony cushion scale. If you're not looking at bugs every day, you're probably going to think it's the same thing when you see it. But if you smush it and it's purple, call Josh. If you see something out there that's a mealybug or scale-looking thing, you smush it, it's purple, give him a call immediately because that's a problem and we need to get it under control. And you'll have to report it to the state, unfortunately, but the Florida Department of Ag has actually been really great to work with on this bug. They make a lot of honeydew, so you see a lot of city mold. Like I said, squish test, turns dark purple, give him a call, he'll give me a call, we'll help you out. The damage, so these are some pretty extreme examples here, but this was a young tree that was under an IPC, and the grower had not been scouting. He put the bags on, put on a regular drench applications, and came out one day and about had a heart attack and called the IPC guys and said, "You killed my trees." IPC guys called me, we went out there and scouted, and it was Levick mealybug all over the place. And so it basically causes massive dieback at the points of feeding, and then they also can get down into these wraps and overwinter in there. So they can cause a lot of damage if you don't catch them. And then this is a mature tree inside of a citrus under protected screens. So those of you who haven't been to Florida and seen our screen houses, we have these massive screen structures that we're growing mature trees in, and we can actually produce some fresh fruit that are quite nice, aside from the bugs. And the bugs are really loving them. So Levick mealybug got into this particular greenhouse. Actually, it's in most of the cups houses I work in, and it was taking out huge sections of this tree. We actually took that tree out and burned it because we didn't want it to move throughout the rest of their house. Because the level on the other trees we could manage, but that was gone. That we could not manage. And it's an emerging like it's one of those things, we've had it for a couple years, but we still don't have a complete answer on it. We've got a lot of data. If you wind up getting it up here, I'll work with Josh and Apurba and Jake to help y'all out. Just please keep an eye out for it so we can squash it before it becomes a bigger issue. It also loves cotton, so if you have any friends that grow cotton, you might let them know. And here's the biggest problem with our fruit. So if it infests at different points of fruit development, you're going to get some variation in your damage. So obviously, that fruit is toast. That's not going anywhere. They literally look like acorns and drop off the field when they get done with it. I call this my pancake fruit. They got that one pretty young in the expansion period. Later on in the expansion period, you used to get less damage. And you can actually still run this through a juicer once it's cleaned up, but most of the processing places are just going to chuck it. And then once it's complete, they don't actually cause as much damage to the fruit, but nobody's going to harvest that. That's a problem. Yeah, it's gross. It's been a really interesting bug to work on, but it is a very challenging bug to manage. So like I said, if you see it, you think you see it, please give Josh a call, and we will work with you. I'm sure some of you guys have seen these. This is Blumulus bonariensis. It is from the West Indies, and it was first found in Florida in 2009, but it wasn't really a problem until a couple years ago. And for the most part, it seems to be consuming the decaying vegetation under the trees. They do move up into the canopy as the tree warms, and it really freaks people out when they see this because they're like, oh my God, they're crushing over my tree, they're gonna kill it. For the most part, if your tree is healthy, they don't do a whole heck of a lot of damage to that trunk, they just climb up it. Where they do see some problems is clogging your emitters. You can see them up over here. When they clog your emitters, you're not getting the water you need. So, if you have a microjet, you have this nice little circular dispersion pattern trying to get the water to your roots. They clog it up where all you get is a trickle right at the base of the emitter. That's not good for your tree. And then they also, the juveniles are born without a shell, and they can actually go into the heads on the emitters. And so we've had to clean out some juveniles from the it's kind of, a paperclip works wonders. The grower figured that one out. Actually, I can't take credit for that. And then we actually do see foliar damage when they get in IPCs. They get up in the IPC because they're getting up off the hot ground, and they get stuck. They don't want to feed on citrus. It's not like their favorite thing on earth, but they get stuck and they feed because you got to eat. So something to keep an eye out for. We're still in the very early stages of understanding how to manage this pest. Sorry, I thought I had another slide on it. If you are interested in this, if you're dealing with this, I'll be talking about some of our research at 3 o'clock in the veggie session, or you can just ask me later. Again, very early stages of research. There's lots of people throughout the Southeast working on this because it likes a lot of different crops. And even though I think we're probably less impacted than some of the other people, we have the money to work on it, so work's happening in my group. And we're just sharing it. Then Diaprepes. Diaprepes has made a massive comeback in Central Florida, and I know that it is in Georgia, and I know that it is in North Florida. This is part of a root weevil complex. This is just our biggest, most voracious root weevil that we have in Florida, and it's highly damaging. So the adults, they're pretty big, actually. I mean, for once, you get to work on a big bug, which is, as an entomologist, exciting. Everything we do seems to be very small, so I got something big. What's kind of concerning there is that each female can lay up to 5,000 eggs, so they make a ton of offspring. And so when they lay their eggs, they lay it between 2 leaves, and they kind of glue those 2 leaves together, so you don't necessarily see the eggs in there. And then once they hatch, the larvae drop down into the soil, burrow down, and start feeding on your feeder roots. Then they'll work their way up to the crown. And then you can see some of this damage. Not only do you have Diaprepes damage, you then get Phytophthora moving into those holes. So it becomes this whole complex of Diaprepes and Phytophthora, so you have to manage for both of them. Especially if you know you have Phytophthora in your field, Diaprepes are just doom and gloom for you. Another big challenge is that they have a huge host range. I know they affect blueberries up here really big time, so don't be surprised if your blueberry neighbor has Diaprepes if they're coming into your citrus field. And we are doing some updated research on these because they haven't been really worked on since before Asian citrus psyllid showed up. So a lot of the recommendations, the chemistries aren't even available anymore. So we're working on updating all of our chemical recommendations. We'll have new recommendations in about a year because we need 2 years of field data to actually make those. There's some of the other impacts of the root weevil feeding. So like I said, they feed on your roots. This had absolutely no feeder roots left. They got up into that crown and they did a number on that baby. And they do consume a greater quantity of roots than the other root weevils that we have in the system. So we have like 4 different root weevils, but these are our most challenging ones to manage. And As they grow in size, they move closer and closer to that crown, and they cause more and more damage. And you'll see those big, deep grooves. And like I said, entry points for Phytophthora, not good. So that becomes a larger management challenge. I'm just going to keep going because Jake's not here, if that's all right. It's like 3 more slides. All right, so again, I mentioned the diaphragma's life cycle. I really just want to show this to y'all in case you haven't seen it. Newer pests to deal with. Adults lay the eggs, those little bitty eggs in the leaves. You probably will never see those eggs unless you're really looking hard. The neonates, or the first instar hatchlings, are super tiny. Probably never going to see them. They go straight into the soil, and then they start feeding on your roots, pupate in the soil, come out, and start doing the cycle all over again. And so we have 2 major periods of adult activity in Florida, normally, April to May, and then again later in the fall. And it's thought that those are actually tied to periods of rain. But it varies throughout the regions, and we're actually right now in the middle of a 3-year trapping program to revise those trapping periods because they don't seem to be as consistent as they used to, probably due to some changes in plant physiology. And so to develop effective management for these, you got to look at the population cycle and that can be figured out with the trapping. And include both cultural, biological, and chemical controls. Again, we'll have some new recommendations pretty soon. And one thing I just really wanted to bring home on this, though, is that we have lots of pests, and you do need to think about them as far as everything you have in your field. What can I get the most bang for my buck on each application that you're going to spray out there? This is actually available online, and I can send you a PDF if anybody's actually interested in how we are looking at this in Florida. This came out last year, and it will be updated as we learn more and get new pests in the system. Okay, I swear I'm done. All right, I have like 2 minutes for questions.
All right, and then don't leave after questions. The Georgia Citrus Association might give a update of what we got going on. So, questions, anybody?
And if you think of them later, oh, I guess my email's not on that slide. Huh, it was on my first slide. Yeah, if you think of questions later, you can email me.
And if don't have any questions, Lindy, you coming to give the announcement, or Mr. Ken is going to give the announcements for the Georgia Citrus Association. Go
I'm
ahead.
going to be very, very brief. Everything I have is in about 5 lines right here. Last year, a Citrus Commodity Commission was formed in the state of Georgia. This is your members of that commission. We've developed a marketing order. The marketing order is for research, education, and promotion. This board's main objective is research. I'm glad to see that we have a specialist now, so we'll try to be able to get some funds to get some research projects going on citrus in Georgia. Some dates that we need to remember is there will be a public hearing on this on February 20th. Voting on this will be March 1st through March 30th. Ballots will be sent out to everybody. Andy Harrison was generous enough to compile us a list. We have that list in the back, but we only have a couple of lists, so please don't take the list, but look at the list, and if your name is not on that list, put your information on there, or contact one of our board members, and we can let you know. Make sure that you get a ballot, and with that ballot, you're going to get a copy of the marketing order, so you'll know exactly what it is you're voting on. And what we're doing in the marketing order is going to be assessed at 2 cents a pound, 0.002 cents a pound, 0.2% per pound, which will, just math it out, 10,000 pounds will cost you $20 to make 10,000 pounds per acre. It's going to be collected by the first handler. First handler will be either the packing house it goes to, or if you're roadside market or whatever, it's up to you to report and pay your assessment. Who will be affected? It's going to be growers that grow or have the potential to grow 50,000 pounds or more. So 50,000 pounds is really not a lot of acreage, whenever you get into full production. So anyway, if you have enough citrus planted to make potentially 50,000 pounds, please make sure that your name is on that list in the back so that we can get you out of ballot to vote.
All right, we're gonna go ahead and get started with our second session here. Dr. Kim Bowman with the USDA Citrus Rootstock Development Research.
Okay, happy to be here to talk to you today. As you can guess by the introduction and what it says on the screen, my work is primarily focused on citrus rootstocks. So part of it kind of compares performance of existing citrus rootstocks that are available in Florida and around the world. The other part, which is actually the biggest part of my program, is developing new rootstocks that are better than what's commercially available, or at least try and develop new rootstocks that are better. Citrus rootstocks are a big deal in Florida citrus production, have been historically. And so the USDA citrus rootstock breeding program in Florida has been going on more than 100 years now. I'm not that old, so my part's relatively newer, although I have been at it about 30 years now. I guess I'm getting to be an old man. I'd like to acknowledge my work has been contributed to by a lot of other people. A few of those are Greg McCollum, Uta Albrecht, Jake Price here in Florida, Hayani BC, and Brandon Page in Florida. But there's actually been a lot of other people that have contributed that I'm not going to make mention of. My research program is supported by USDA base funding, but we also get support, financial and otherwise, from several different agencies, included NEFA and the Florida Department of Agriculture, CRDF, and CRB grants. And support from in various ways from many different Florida growers. So some of the trials that I'm going to mention data about are with grower cooperators in Florida. Well, I think part of the backdrop of all this is, because I'm based in Florida, is Florida citrus production. This little graph shows production in Florida and the U.S. over the last approximately 20 years. You can see in 2001, 2002, production in Florida was something like 13 million tons of citrus fruit, and the total U.S. was something like 16 million tons. So Florida production was a huge proportion of total U.S. production. But we had HLB up here in 2005. And you can see production declined, well, not entirely from HLB. To some extent, that was from hurricanes and citrus canker. But through the following 20 years, there's been a huge continuing decline in production. Most of this continuing decline in production was from effect from HLB disease, Wild Being or citrus greening. And so now Florida citrus production is much lower than it was 20 years ago, something like 2 or 3 million tons of fruit a year. And it's a much smaller proportion of the U.S. production. Of course, Georgia citrus production, I guess, has increased quite a bit. It's still relatively small, but you all have made good progress. And I think it does make sense that you're paying attention to what rootstocks might be best for new plantings. The citrus rootstock provides for a lot of different things. It provides anchorage for the tree and uptake of nutrients and water. Those are all really important things. It has a strong influence on the size of the tree survival, the yield of the fruit, and the fruit quality. So even though the scion certainly greatly controls the fruit quality characteristics of the fruit, rootstock has a big influence on the Brix acid and size of the fruit. Citrus rootstock determines tree tolerance to many of the diseases, or at least has a strong influence, and also tolerance of environmental stresses that affect the root system. So the rootstock affects tolerance of Phytophthora, nematodes, salinity, drought, flooding, and high pH. And the rootstock also has a strong influence of tree tolerance to most diseases and stresses the above ground portion of the tree. So citrus, tristeza virus, Huanglongbing disease, and freeze or cold damage. So this just kind of illustrates the importance of the rootstock for tree anchorage. You don't have as much problem from hurricanes as Georgia as we do, but in Florida we have regular hurricanes affect citrus plantings. This is the result of hurricane damage a number of years ago in Florida, just blowing trees over. So even though the trees had good root systems, extended wind and with a lot of water, at least depending on what kind of root system you have, the tree can just topple over. I mentioned tree survival. So this is a planting of grapefruit at 2.5 years old. The tree on the right is grapefruit on a rootstock called US 812. The tree on the left is grapefruit on the rootstock swingle. You can't hardly see the tree, but if you look in closely, you can see the tree didn't grow well at all. This is in a particular site in Florida where there's, I guess, a combination of a couple different challenges. The biggest part of the challenge is Diaprepes weevil, the previous speaker mentioned. In this area where Diaprepes weevil is present, it's not just Diaprepes weevil itself, but it's the subsequent Phytophthora damage that makes swingle especially badly damaged, and trees fail almost entirely on that rootstock, while other rootstocks, trees on other rootstocks do particularly well. And then I mentioned rootstock influence on tree size. Actually, this is tree size pre-HLB, so maybe it would be more reflective of what you'd have in Georgia now, but Hamlin Sweet Orange on US 802, tree 17 years old in this block, looked this size. Trees on US 897, also healthy and highly productive, were less than half the size. So the trees on US 802 were about 18 feet tall, trees on Hamlin about 9 feet tall, on trees on US 897. So a huge influence potentially from rootstock on tree size. And that affects a lot of different things. One is you choose a different planting density to start with if you know you're going to get a big difference in tree size. Most of the time, growers would prefer smaller trees just to simplify management and harvesting. But certainly larger trees make more fruit per tree as well. So you have to consider that when you're thinking about profitability of a particular planting. Well, as I mentioned, my research program is mainly focused on rootstocks and developing new rootstocks. I would define the major components of the work as first to evaluate field performance of existing commercial rootstocks and identify those most suitable for use in Florida and other parts of the U.S. Create new hybrids and identify those most promising by field evaluation and release new hybrids when appropriate, and develop new information and methodologies that improve or accelerate breeding or commercial use of good new rootstocks. So the first, kind of evaluating performance in existing trials, I'm going to mention data from several different trials. This is a bingo trial. Actually, the picture is of a trial in Fort Pierce. There's a similar trial in another location at the Whitmore Farm. That's where I'm going to show you the data from. But bingo is a high-quality early seedless mandarin developed by University of Florida and released a few years ago that at least initially had a great deal of commercial interest. This data, like I mentioned, is from a trial of that same scion at another location in central Florida. And it just compares performance of, I think it's 13 different rootstocks over a period of several years. The trial was planted in 2018, and you can see this is kind of showing data primarily through 2022, so kind of over a 4 to 5 year period. The rootstocks are listed on the left. Uh-oh. Oh, not sure what happened there. Shoot dieback is shown in those first two columns. One of the problems with the bingo variety is there's a substantial amount of dieback of the young flush, especially when the trees are small. So we rated that over a period of time, and you can see here the numbers are percentage of the shoots that died back, with the ones with the green shading had the least dieback in each column. The ones with the gold shading had more dieback. We looked at canopy health, and this was scored where the higher number is better. So a number of 5 means that canopy health would look essentially perfect, but a lower number means it was a less healthy canopy. Again, green shading means better. canopy volume and canopy height, so that's just giving a reflection of tree size. In this case, the green shading means larger trees. Cumulative fruit number, so that was over a 2-year harvest period, the 2021 and 22 season. Again, green means more fruit, gold means less fruit. Fruit size and fruit Brix acid ratio, so those are both reflections of fruit quality. In general, at least with the bingo variety, larger fruit is better, and a higher ratio in general is more desirable. So those, the best are green. And again, using the shading to kind of give you an indication there, what you're looking for is a rootstock that gives you the green category for most things, probably. US 942 was the rootstock you would pick out here as being the best in pretty much every category. It gave the least dieback, the best canopy health, the most fruit, the best fruit size, and the highest fruit Brix acid ratio. The only thing you might question there is canopy volume. You might say you prefer a smaller tree. In this case, a tree height of 7 feet still isn't too tall. But again, these are relatively young trees, so I guess over time you might decide you'd prefer a rootstock that gave a smaller tree. In this case, it not only gave the most productive and the healthiest tree, it gave the largest tree. That could be a problem, but it wasn't during the course of this experiment. I'm going to mention quickly a trial with Washington Naval Scion, Washington Naval Sweet Orange Scion. Actually, the trees are still young, and so that there's not really data. I'm just going to show you a few pictures. The trees were planted in 2020, so they're now 3 years old. Just to show you what they look like, the US 812 rootstock tree on the left, C146, which is a California rootstock in the middle. Rich 16.6 is a Panseris trifoliata selection that's common in California. I don't know if you would use that in Georgia, but it's going to be very similar to Rubidoux in performance. And again, C22 is another California variety. US 942 is a Florida preferred variety that I mentioned looked the best with bingo. This just shows you what one of the trees looks like with Washington Naval. Swingle on the right. US 1516 and US 897 are two other relatively new rootstocks in Florida that look good in some trials. We also have some commercial interest in growing lemons in Florida. Bear's lemon is one of the most common lemon varieties being used in Florida. These trees were planted in 2018, so they're now about 5 years old. And again, the experimental design, just to show you a little data through the first 2 harvests, 2020 and 2021 season. Like the bingo trial, this is sorted. I guess I didn't mention that with the bingo, but sorted by the most productive rootstock at the top. The rootstocks are showing here on the left. And then you see tree survival, weight per fruit, percent juice, canopy volume. But like I said, they're sorted in this table by cumulative fruit count over 2 harvest seasons. So you can see in this case, US 896, US 897, 812, and 942 were more productive in fruit production than the others like Volcom or Swingle and Standard Sour that would be more common in commercial use. Just going to quickly mention my work with Jake Price in Georgia with an Owari Satsuma trial. I'm not going to show you any data from that, but this is just a photograph of one of those, the trees in the trial on US 942, one of the rootstocks that had done well in the trial. The trees were planted, 8 to 9 years ago for the most part. I'm supposing Jake has presented data on this trial sometimes previously in Georgia, but in general, the trees have done well, and it's a really well-done trial. I'm happy with the work Jake has done on that. Well, most of the fruit grown in Florida for commercial production is for citrus sweet orange juice. And this is a relatively large trial that was done with Valencia sweet orange in Lake Wales, Polk County, Florida. The trees were planted in 2008, and data was collected over a period of time. The trees were 100% infected with Liberibacter during the course of the trial. Nevertheless, the trees did really well and were highly productive, at least on current standards. The data shown here is over a 4-year harvest period. Like the other trial, this is only showing yields in pounds of fruit per tree. The rootstocks you can see on the left there, again, it's sorted with the most productive rootstock at the top. And you can see in terms of total pounds of fruit per tree, Swingle made about 400 pounds of fruit per tree over the 4-year period. US 942 was the most productive with about 500 pounds of fruit per tree. So now I'm going to kind of transition into mostly talking about new rootstocks, or the part of the project that's developing new rootstocks. 16 rootstocks have been released by me so far from the USDA breeding program. You can see them here, the four at the top, US 812, US 802, US 897, and US 942 are the ones that have a rather large amount of commercial use in Florida now. The 5 at the bottom that are identified as super sours are very new releases and don't have a lot of commercial use yet, but I think have a lot of potential for the future, and I'll mention more about those coming up. But first, to kind of highlight the commercial use of those that were released previously, the Citrus Budwood Report, the most recent report that's been published, the 2021-22 harvest season. You can see the top 10 rootstocks there. US 942 was the most used rootstock currently each of the last 5 years, with about 1 million trees propagated every year on that rootstock. And you can see it was also the most used rootstocks each of the previous 3 years as well. US 812 and US 897 are pretty heavily used, but nothing like US 942. And other important rootstocks in Florida recently are Kuharski, X639, Swingle, and standard sour orange. In Florida, Huanglongbing disease really is the major factor, I think, affecting citrus production, highly damaging to all citrus crops. We do know from evidence in trials and experiments, tolerant rootstock improves tree performance with HLB, but even the best rootstocks suffer significant damage from that disease, so we need better rootstocks to help tolerate the disease even better. As I mentioned before, the Florida industry is heavily focused on sweet orange juice production, so most of the trials that I'm conducting are with juice sweet oranges. I have now about 20 replicated sweet orange field trials with about 400 different new hybrid rootstocks, and we're using that information from those trials to help identify new rootstocks that are even better for commercial production. Generally, I'm including 4 standard rootstocks in these trials that are so we can cross-compare performance in different trials. Swingle and standard sour orange are 2 rootstocks that are still real common in use in Florida for commercial production, so those are the primary standards for comparison. Cleopatra mandarin is another common citrus rootstock. Ridge sweet orange is included in the trials kind of because it's sort of an equivalent to what sweet orange would do at its own roots. Ridge is a sweet orange selection that was made specifically for use as a rootstock, but it's essentially pretty similar genetically to sweet orange. So it gives us a comparison of tree performance on its own roots. This is just a partial list of the sweet orange trials. You can see these are all with Valencia or Hamlin sweet orange scions. In total, these trials have about 9,000 trees that we're collecting data from every year. I'm going to focus now on a couple of different trials just to give you an idea of what the data looks like and the more better performing new rootstocks. This is with Valencia scion. The 2 trials were planted in 2014 and 2015 in St. Lucie County. The combined trials have 101 different rootstocks planted with 12 replications of each. And again, those 4 standard rootstocks that I mentioned before are included in those trials. This is what that area looks like from the air. And the 2 trials are shown kind of within the circle there. This photograph was taken when the trees were about 5 years old, 4 or 5 years old. And this is super sour number 3, one of the new rootstocks that was released, compared to what trees on standard sour orange looked like in the trial at the same time. This is what trees on Swingle looked like at the same time, trees on Cleopatra at the same time. More recently, actually, I think this photograph is a year old, but it's what it looks like more now. Ridge orange, which I mentioned, is the equivalent of a sweet orange on its own roots. It looks like that. On super sour number 4, that's more like what the trees look like. So now that the trees are 8 or 9 years old, there's a huge difference in relative performance of trees on the different rootstocks, in some cases pretty striking differences. If you look at performance, and in this case it's crop fruit production over a 4-year period from 2018, 2019, 2020, and 2021. So this is cumulative fruit yield in pounds of fruit per tree. Over that 4-year period, performance of standard sour orange rootstock is there. So again, this is with Valencia scion, kind of in the middle of the pack in terms of performance. Cleopatra and Ridge orange there. Swingle, I was actually a little better than standard sour orange. But the super sour number 4 and super sour number 5 are there. So kind of at the top of the pack of the, and this is, I think, about 60 rootstocks included in this comparison. This is a comparison that includes more rootstocks in 2 different trials combined. Each balloon here represents the mean values for all trees on that rootstock in the trial. And in this case, it's presented as a relative performance, not an actual value number, with higher fruit yield is the balloons to the rootstocks with balloons to the right. And those that have thicker, healthier canopies higher up on the chart. And the size of the balloon indicates the yield efficiency, so the amount of fruit per canopy area. So larger balloons is kind of a better thing too. In this case, the relative comparison of everything is to standard sour orange, the pink balloon in the middle. So it's showing as a 0 relative canopy yield on the metric on the bottom. So everything to the right has a higher fruit yield than standard sour orange. And it's showing as a 0 on the relative canopy health metric, the up and down. So everything above the pink line is a healthier tree than trees on standard sour orange. And looking at it that way, sort of everything in that green circle in the upper right corner is what I'm calling the super zone, kind of what had really superior performance in these trials. Looking at those other standard rootstocks where they are, where their balloons are here, that's Swingle, Cleopatra, and Ridge orange. So you can see if you'd planted Valencia in this location or in Ridge orange or on its own roots, it would have performed very poorly. Not really a surprise, but people still talk about growing citrus on their own roots. Generally not a good idea. The rootstock Super Sour 4 and Super Sour 5 are there on the chart, Super Sour 3 there. So you can see in terms of cumulative yield, in these trials, Super Sour 3, 4, and 5 considerably outperformed the standard rootstocks in terms of yield, and at least 4 and 5 looked like a lot healthier canopy on the trees as well. Looking at a different set of data from a different trial, cumulative fruit yield, you can see comparing Super Sours 3, 4, and 5 to standard sour orange and the other standards. Cumulative yield here, Swingle, Cleopatra, Ridge orange, Super Sour is 3, 4, and 5, as I mentioned here, all with higher cumulative fruit yield over in that case, it's a 5-year period. And again, the Ridge orange is like sweet orange on its own roots. Looking at canopy health score, I mentioned is another metric. We're taking the standard rootstocks compared to those 3 super sours all look better. Looking at tree height, you can see standard sour orange and Swingle tree size here. And this is meters. So tree size was for the Swingle standard sour orange and the three super sours shown here are all actually pretty similar, roughly 7 to 8 feet tall. And this was when the trees were 8 years old. The rootstock super sour 1, 2, 3, 4, and 5, the 5 that have been released also have demonstrated, and I guess you all are not that familiar with maybe Florida production areas. The other data I was showing you was what we call flatwoods, East Coast flatwoods area. We have the other big production area within the state is ridge, what we call ridge sites, so it's higher elevation, better drained soils. These 5 rootstocks also have had superior performance with Hamlin, which is a different sweet orange grown in Florida on the ridge site. And just to give you a quick example of that, Whitmore Hamlin trial here comparing standard sour orange, Swingle, Clio, and Ridge to super sour number 4. You can see out yielded considerably over the 3-year harvest period. And this is comparing super sour number 5 to the standard rootstocks as well. And this is actually a different trial at the same location with sweet orange. And there's a recent publication that gives a lot more information about relative comparison of the super sour rootstocks. It's available. If you all want more, I can. If you can either find that or I'd be happy to send it to you if you send me an email. So another thing I'm going to touch on briefly is developing of information and methodologies that improve citrus rootstock breeding and commercial use. Genetic uniformity from seed is a trait with significant importance in citrus rootstock use. However, I think recent advancements in technology to propagate citrus rootstocks by cuttings and tissue culture give us methods that mean we don't really need to be using seeds for rootstock propagation in the nursery anymore. One of the publications here was from several years ago, but basically if you use the right methods, this is cuttings about 3 weeks after the cuttings were made. You can see every one of those is growing wonderfully. And it's just using a single node cutting from the shoot of the tree. You put them in soil, and within a few weeks you have wonderfully growing plants with that you can use as liners without having to mess around with seeds. Probably seeds is actually easier and quicker. It's a little less specific methodology needs, but we don't have to be restricted to just things we can propagate by seeds anymore. And I know there was concern in Florida, maybe still is, and maybe in nurseries in Georgia as well, that if tree performance is going to be equivalent if we propagate by cuttings or tissue culture. Some work that I've done with Dr. Uta Albrecht at University of Florida indicates that the tree performance is pretty much the same by all 3 propagation methods, seed, cuttings, and tissue culture. Where we've grown trees in the field, dug them up, looked at how we look at performance also, but seeing how the roots look. And in this case, this photograph, the plant tree on the left was a seedling, the one in the middle was from a cutting, the one on the right was from tissue culture. So we've kind of looked at it in detail. It seems like the rootstock genotype has a huge effect on tree performance. The way you propagate the rootstock liners doesn't have much of an effect. And the other thing I was going to mention about the program, a big focus for now and the next few years is to use the replicated data from these trials to genetically map important rootstock traits and develop molecular markers that can help guide future rootstock breeding. A lot of this work is being funded by NEFA, grants from USDA NEFA. And we have the first publication that kind of describes that methodology now, where we are, again, identifying specific parts of the genome that we can select for in the breeding process. So we make new hybrids. We can analyze the genes with sequencing and determine which new genotypes look the most promising right before we even plant the trees in the field. We still have to go through with planting trees in the field and seeing how they perform over time. But rather than planting out 400 different rootstocks, only which 5 are going to be good, we can pick out the 5 that are probably going to be the best to start with, and we can put a lot more effort into the most promising rootstocks from the beginning. Depending on the parental combination and the frequency of new rootstocks that have good combinations of tolerance to the disease and high yield and high fruit quality is about 1 to 2%. We are using performance in the trials to map the important traits and develop selectable markers that will help guide. So the idea is to make trees for which the rootstock is going to really give us super performance. So that's all I had planned to say. I'd be happy to answer questions if there's time. Any questions? You can always send me an email. Yes, sir.
So you found that the propagation from tissue culture in the field is no different than using the seeds and all that?
Yes, that's basically the result. I mean, there is a lot of variability in the field, regardless of how you propagate.
Right.
Quite honestly, it is apparent in some of the work that there's actually several publications we've had come out with this. There's still ongoing work. There is a really big impact on tree performance in the field by nursery, the way the trees are produced in the nursery. So if they're being grown in pots, what size pots are being grown in, whether, and in the nursery.
Tall pots.
Yeah, tall pots, fat pots, what soil you put them in. Usually you start a seedling, like if you're growing a seedling, you start it in one container and then you put it into a different container to grow it out. So a lot of the details about that nursery production does have a really big impact on what goes in the field. And that is interrelated with whether it's seedlings, whether it's cutting, or whether it's tissue culture. And you certainly can do things in the nursery by any of those propagation methods that's going to mess up tree performance in the field. If you do things that stunt the roots or create a J root in the nursery, or somehow don't allow a good root development, that's going to really have a negative impact on field performance. But it's not because of the propagation method initially. It's what you did with it in the nursery as you were growing your tree. So to answer your question, no, it doesn't seem like whether it's seed, cuttings, or tissue culture itself makes a difference on tree performance. But some of those other things you do with the tree in the nursery can really hurt tree performance in the field. Yes, sir.
Have you had a chance, specifically with the super sours, to kind of look at them in Clinton or Georgia to see the cold tolerance out of compare?
I have not yet. As far as I know, there's one case, I believe, where Jake, I wish Jake was here, but I believe Jake told me one of the super sour rootstocks that he had gotten, and it was super sour number 2, that he had gotten seed and trees propagated 2 or 3 years ago. Do you by chance know? 3 years ago, and I believe he told me even though he expected the trees were going to be highly cold tolerant, they were not. They'd had a lot of damage in a freeze you had last year. Do you know the details of that? Sorry, maybe you know better than me.
Damage, and the super sours, we know it's cracks in the bark from the hard freeze from that December time, I believe, last year. We noticed that there was some damage to the seed.
But I believe that was super sour number 2 is what he told me. So at least that one, as I understand it, the current, even though, well, I'm not sure how it was, like the age, what you can compare it to, because I don't know if you had other trees like the same age and everything to compare, but at least it didn't look encouraging for cold hardiness, the result of that. But as far as I know, the other 4 that have been released have not been tested yet in Georgia. I mean, I'd be happy to see them tested or work with somebody to do it, but we just don't have any information yet.
Quincy would be, it's pretty cold in Quincy.
That's true, absolutely. Yes, sir, in the back.
As you develop these super sour and others, have you got a test that you use just to see how susceptible to Tristeza and some of the other viruses?
Well, most of my program, we have done over the years a lot of different specialized testing with things like Phytophthora, Tristeza, and HLB in particular. To some extent nematodes, but focus testing on particular diseases is really challenging, time-consuming, and expensive. So mostly we don't test individual rootstocks against these different things. What we mainly focus on is planting trees in the field that have these problems and then observe how they perform in the field. But normally the problems aren't one thing alone. It's a combination of several things at once. So for example, I showed the photograph of the trial that was affected by Diaprepes. But it wasn't just Diaprepes, it was Diaprepes and Phytophthora, and it was in a soil that's particularly prone to flooding as well, so really wet feet. And that particular trial was actually done before HLB was a problem, so we didn't really have HLB as a, and to answer, like for Tristeza, because we have Tristeza endemic and widespread in the areas where I'm growing these trees, and the same with HLB, every tree becomes infected. We don't inoculate, we basically plant the trees in the field, grow them out over a period of time. We do monitor spread of the disease, the Tristeza and HLB, or Liberobacter is the name of the disease that causes HLB. So we do monitor the spread of citrus Tristeza virus and Liberobacter in the trees over time. So for the data that I showed from the trial in Fort Pierce, for example, when the trees were 8 years old, 100% of the trees are infected with Liberobacter, the bacteria that causes HLB at that time. 90% of the trees are infected with Tristeza. So we don't, but they have both Tristeza and Liberobacter, both in the tree. So we can't really say which is a result of Tristeza and which is the result of HLB, but we can say if a tree performs really well, it must be relatively tolerant to both. So that's kind of the comparison that's typically happening. So we don't have information that says specifically to Tristeza. I have more information specifically on HLB because we have done a lot of greenhouse studies where we inoculate under controlled conditions. They don't have both. Another thing I guess I'll mention quickly, I've kind of forgot to say something about before, but all of these trials were established without using individual protective covers, without using reflective mulch, without using Carolyn. So none of that management was done with these plantings, these trials. These were all done kind of with the intention of seeing what tree performance would be like with these rootstocks, where they were kind of having to fight the diseases on their own without any special methods. Presumably, if we established the trees with protective covers or used other methods to decrease spread and severity of HLB disease, maybe they would do better. I do have new trials that I've planted in the last year or two where we've used IPCs in the establishment, and some trials where we're injecting with oxytetracycline, as is standard commercial practice in a lot of places in Florida now. So we'll have good information about what happens if you use the best rootstocks along with the best management practices. We don't really know for sure yet, but I guess the presumption would be if you have good performance without those extra measures, probably you'd have really good performance as long as you do the other things as well. But all of the advantages from a better rootstock are advantages you get without any special management at all, I guess. So I'm just saying the rootstock doesn't cost you anything to use the best rootstock to start with. If you plant the best rootstock, it's going to help you out no matter what happens, kind of at no additional cost. Any other questions? Thank you very much.
Hold on a second while our Citrus Commission come up here and give a few updates, what they got going on.
I'm Jamie Patrick. I'm with the Citrus Commission here in Georgia. Thank y'all. I just want to tell y'all we do have an annual conference February the 27th in Tipton, Georgia. These are the members of our commission. If you have any questions about this, any of these guys can answer anything that you may want. We have a goal with the commission is we're trying to support research, promotion, and education. We do have a marketing order that's coming in that we are looking at trying to have an assessment. That assessment is approved by the producers here in Georgia. So if you are a producer here in Georgia, please sign up in the back if we don't have your information so you can get a ballot. You would have to approve this for the assessment to happen. The assessment would be 0.2 of a cent per pound. A producer is 50,000 pounds or more. So if you have less than 50,000 pounds, the assessment would not apply to you. If you have more than 50,000 pounds, then it would apply. We are having a public hearing in Tipton at the Nestball building February the 20th at 1 o'clock in Tipton, Georgia. We will be voting. We'll be sending ballots in the mail, and the voting will be March the 1st through the 30th. So if you would, please consider voting yes for that marketing order to help us support the research, promotion, and education of citrus in Georgia. Thank you.
That wraps us up for the morning session. Y'all feel free to go to the trade show and grab a bite to eat. We'll start back at 1:30.